About the inheritance of complex traits. And we're going to talk about a quantitative variation. And to talk about that, we are going to talk about two concepts that are the mean, the variance, and standard deviation in the populations that we're interested on. Then the genetic model that we have to understand, the basic one, and genetic and environmental variances. And at the end, we're going to talk about the heritability. And there are two types of heritabilities, that it's the broad and the narrow sense heritabilities. So remember that when we discuss about a simple model or a simple allele model, we mentioned that when we have only one locus that control the trait, we can have only have three categories. One for the recessive homozygous genotype, another one for the heterozygous, and another one for the dominant homozygous genotype. So in that case, we are going to have just three categories if the trait that we're interested on is controlled for only one locus. And each time that we have more locus or more loci that control the phenotype, we're going to have more categories of the phenotype, until one point that we have a lot of loci that control the phenotype, and we're going to have a continued distribution of the phenotype in the population. So basically, when the traits that show a continuous range of variation like this one don't behave as a simple Mendelian fashion like the one locus model, that kind of traits are known as a quantitative or complex traits. And that is the area when quantitative genetics is important. So basically, quantitative genetics applies statistical methods to understand the inheritance of these kind of traits. And there are some basic concepts that we need to remember, and one is the mean. So most of you are familiar with that, and to estimate the mean in a population, we just have to add all the phenotypes or the data, the mean of anything. We just have to add all the data that we have, and we have to divide it with the population size or with the total number of samples that we have. And that it's a straightforward way to estimate the mean. And another way to estimate the mean is if we can take into account the frequency. So here in this table, so first in the formula, we have that the x with this line on the top represent the mean of the population. And here in the formula, you can see that this f represent the frequency of the phenotypes that we are checking in the data or the data points, and the X represent the number of classes or data points that I have, different data points that I have in the population. And just to show you how that formula works, I have here in total 100 individuals, and we collected the phenotypic data of all the individuals, and this count is the number of samples in the population that have this specifically this phenotype. So for example, for 100 we have only 100 one individual that have 156 centimeters of height in this data set. And then you can see, for example, we have two individuals here that have a 157. So as we know how many samples in the population has the phenotype of interest or the classes or different classes, so we just have to multiply the frequency of that phenotype and the phenotype for itself. So here, for example, in the first case, we have that only one individual in 100, that is the frequency of 0.01, has a phenotypic value of 165. So we have to multiply that value, and then we have to add all the other classes with their frequency, and at the end we're going to have the mean of the population, that is 170. So that it's kind of tricky if we have all these different classes, but in quantitative genetics is pretty useful, basically because if we're interested just in one allele, in one locus, we can have three different categories. So here we have the homozygous B1, the heterozygous, and the homozygous B2. And we know what is the frequency, that is the genotype frequency that we learned to estimate in the previous video. And we have the trait or the phenotypic value of each genotype. So as you can see here, I just have to multiply the frequency times the trait of the phenotype, and that is the contribution to the mean of every single or each single genotype. And at the end, if we add all the contributions to the mean of every single frequency and value, we're going to have the mean of the population. So it's pretty useful in quantitative genetics. And another two concepts that are important in quantitative genetics are the variance, that basically measure how far a set of numbers is spread out from their average value. So here you can, and is represented with an S square. And basically here in the formula, you can see that this xi represent each data point in our data set, and we have to subtract the mean of the population that it's represented here with the green line, and we have to square that difference. And this symbol here represents that we have to sum or to add the difference of every single data point and the mean and square that value. And then we have to divide that product with the population size that is represented here with n, and the -1 is just a way to try to correct a bias caused because the small size of the population. So it's usually we just subtract 1 to the population size because we're not working with all the population of, in this case, the dogs. And we have also the standard deviation. So the standard deviation is just the root square of the variance. And the difference between them, both of them measure the variation of the data, but the standard deviation is kind of easier to interpret because it's in the same units that the original data set. So as you can see here, we root square because here we square the values of the difference between each data point and the mean. And basically we have to do that because if we don't, the positive values are going to cancel out with the negative values here. So that is why we need to work with positive numbers. And here we square root the data, the variance, and with that we transform back the difference to the same units that we were working on in the data set. And now we can talk about the genetic model. So this is the simplest genetic model, that is the phenotype is equal to the population mean plus the genotype plus the environment. And you can see here that as the population mean is a constant in the population, so we just can remove the population mean, and we're going to have that the individual's phenotypic deviation of every single sample is equal to the genotype plus environmental effects. And the phenotype, it's basically the product of the genotype and the environment. So that is why it's important. That is why we're interested in to understand the genetic and the environmental component. So as we're interested in the genotype and in the environment factors to understand the phenotype, we have to understand the variance of these factors. So the phenotypic variance is equal to the genetic variance plus the environment variance plus the covariance between the environment and the genotype. But as in plant breeding, we control the experiments in the field. So basically, we can plant different genotypes, or the genotypes are placed into different environments and at random. So that is so important that we have to use a random distribution of the genotypes in the field. So the covariance between the environment and the genotypes in theory it's zero. So that is why we have to remove that, and now we have the variance of the phenotype is equal to the genetic plus the environmental variance. And that it's the way that the formula is a little easier to understand. We're going to talk about the heritability, that basically is the proportion or the variation in the phenotype that could be explained with the genotype. And there are two types of heritability, that is the broad and the narrow sense. So basically here again we have the formula that the phenotypic variance is equal to the genetic plus environment. So we're gonna focus our attention in the genetic variance, and the first variance, or the first heritability, is the broad sense, that is represented with the uppercase H and the square of the upper case H, and is equal to the all the genetic variance, that is this value that we have here, over the phenotypic variance. But here it's something important that you have to remember, because the genetic variance it's equal to the additive variance plus the dominant variance plus the epistasis between the loci in the genome. So the additive variance is represented here, and we have or a gene or a locus has an additive effect when the heterozygous lines are just in the middle between the two homozygous. So here, as you can see here, the recessive homozygous genotypes have a value of -1, the heterozygous have a value of 0, and then the dominant homozygous have a value of +1 or +1. So you can see here that the heterozygous are just in the middle. That it's an additive effect of a gene or a locus. Then we have a different kind of dominances or dominance effects. So here we have the typical complete dominance, that it's that the heterozygous genotypes have the same phenotype that the dominant homozygous genotypes. And this color here, the red color, represent the deviation in the population that is due to the dominance. So here you can see here that there is a partial dominance, and this partial dominance means that the heterozygous lines are not in the middle, are closer to the dominant homozygous genotypes, and the D represent the dominance, and again the red color represent the deviation that the dominance has if we compare that alleles with another one that have a complete additive effect. And we have other kind of dominance, that it's a over-dominance, for example, that is the hyper-vigor, when we are talking about hyper-vigor, and is when the heterozygous lines has a more extreme value than the homozygous dominant homozygous genotypes. And we have the underdominance, that is the same but in the another direction. So the heterozygous has a lower value than the recessive homozygous lines. So as you can see here, there are different kind of dominances, and at the end, and that variance between those kind of dominance, it's taken into account in the dominance variance right here in the formula. And then we have the epistasis, and the epistasis, it's basically the interaction between genes. So easy example to show you that, it's in mice. So there are two genes, with each one with two alleles, and as you can see here, the combination of those genes produce three different colors in mice. And that it's important because, as you can see here, this is a very simple example because there are just two genes, so it could be the interactions could be bigger, could be involve several genes in the genomes, but here you can see that interaction between genes could be complex. And the variance of that epistasis is taken into account in this variance in the formula. So as you can see here, or why it's important to have a understanding of the broad sense heritability is because every single genetic variance is included into the broad sense heritability, but that variance or that heritability is not transmissible to the next generation in a predicted way. And basically because here, remember, and again in meiosis, remember that the parents have to produce gametes to the next generation, but they just produce haploid gametes. So that means that they produce just one of the alleles, or they transmit just one of the alleles that they have, and so they don't transmit all the genotype that they have. So what I mean that they are not transmissible to the next generation is because they cannot transmit the dominance or the epistasis because they just transmit one of the alleles that they have in the genome. So for example here, I'm crossing one yellow line with a green line, and the yellow line produce just one gamete because they are homozygous, so produce just a big Y a gamete, and the green one produce the small Y gamete. And the heterozygous lines are yellow, so that means that the big Y is dominant, and all the lines that are heterozygous are going to present the yellow phenotype. And if just think that we're working in common being here, so it's we can advance for self-pollination this F1, so that means that this genotype could produce two different gametes, so the big Y and the small y. And when I cross, or when I advance one generation, I'm gonna have a 75% of the lines of that progeny are gonna be yellow, and just 25% are gonna be green. And the issue with that is that the proportion of the phenotype, it's 3:1. As you can see, 75% of the phenotypes are yellow and just 25% are green, but the genotypic ratio, it's different, it's 1:1. So again, that is because the parents transmit just the alleles to the next generation, but not the genotypes. So here, as you can see, there is a difference between the genotype ratio and the phenotype ratio, and that it's why it's challenging to predict in a easy way the broad sense heritability. So that is, it's difficult to predict the dominance and the epistasis. So that is why it's inbreeding is most interesting to talk about the narrow sense heritability that just take into account the additive variance, and we have to divide that with the phenotypic variance. So remember that the phenotypic variance is the genetic variance plus the environmental variance. So here The phenotypic variants include the additive, dominant epistasis, and environmental variants. And the good thing with the narrow sense heritability is that it's transmissible to the next generation in a predicted way. So just imagine that we have these two cases. One gene that have an additive gene action and another one that have a dominant gene action. So just I'm going to show you the first example. If we have some gene that it's completely additive, we cross two lines that have three flowers. So as you can see here, to have three flowers, the genotypes must be homozygous for the B2 allele. So both of these two plants are homozygous for B2. And when I cross these two lines, all the progeny is going to have three flowers as well. So it's easy to predict the progeny when the gene action of the when we have an additive gene action. But in the other hand, if the action is dominant, we can cross again two plants with three flowers. But in this case, just imagine that the two plants are heterozygous. They are heterozygous and have three plants because the B2 allele is dominant, and that basically give the phenotype of three flowers into the two plants. And when I cross those two parents, the progeny, 75% of that progeny, it's gonna have three flowers, and 25% is gonna have just one flower. And if in the previous example, like all the plants presented three flowers, the mean of the population of the progeny was 3. But in this case, although both parents have three flowers, the mean of the population is 2.5. So as you can see here, because the dominant deviation, it's difficult to predict the phenotype in the offspring. And another way to see the heritability in narrow sense, it's with a typical example of to do the linear regression between the offspring and the mid-parent value. So basically, the regression between the offspring and the mid-parent value, it's equal to the covariance between the mid-parent value that it's here in X and the average or the mean of the offspring of the cross of each cross between the parents. And we have to divide that over the variance in the phenotype, and that is equal to the additive variance over the phenotypic variance, and that is equal to the heritability. So as you can see here, it is possible to estimate the narrow sense heritability doing regressions between the offspring and the mid-parent value or the parent values as well. And to use again the example that we just mentioned about the plants, imagine that we have two homozygous lines. So we have a homozygous B1 that have just one flower and homozygous B2 that have the three flower. As both of them are homozygous, they just can produce one gamete. So in the B1 homozygous just can produce the B1 gamete, and in the B2 just in the homozygous B2 just the B2 allele. So that means that all the population is going to be heterozygous, and all the population or the progeny of that cross is going to have two flowers. And as you can see, it's easy to predict when the phenotypes are, when the gene action is additive in the population. So as a review, Again, remember that quantitative genetics basically applies statistical methods to understand the inheritance of quantitative traits. Uh, Try to remember or to understand why use the frequencies to get the mean of the population. It's important. It's going to be important when we're going to talk about genomic selection, because it's a simple way to understand the expected mean of the population, and also the variance. So remember that we are going to talk a lot about genetic variance, and specifically about additive variance. And the genetic model is kind of simple, so we have to understand what is the genetic component plus the environment that influence in the phenotype, and remember that in theory the complete formula include the mean of the population, but like the mean of the population is a constant, we don't need to include that mean here. And then remember that we have two kind or types of heritabilities: the one that is in a broad sense, that include all the genetic variation in the population, that include the additive dominance and epistasis, but as dominance and epistasis depend of the genotype, so to have the two alleles and the interaction between genes in the population, so that a kind of heritability is not transmissible to the next generation. And then we have the additive variance, or the narrow sense heritability, that include the additive variance, and that heritability is the one that we are most interested, because that is the heritability that is transmissible to the next generation. This is the end of this video.